of two-day DBT-sponsored National Conference on Current Landscape on Delivery System for Effective a Global Perspective. Today we will have three scientific sessions. For first scientific session, we have with us Dr. Ambar Vyas. I welcome sir on behalf of Columbia Institute of Pharmacy. Welcome, sir. Dr. Ambar Vyas. Yes, sir. We are getting your voice.
Good morning, Amber sir.
Good morning, Rajnikant.
Sir, your voice is not audible clearly.
Is it clear now?
Now it's clear, sir.
Okay, sir.
Okay.
Dr. Ambar Vyas is presently working as Associate Professor at University Institute of Pharmacy. Pandit Ravi Shankar, Chhattisgarh.
रजनी सर प्लीज म्यूट कर लीजिए अपने को.
Sir, I request you to kindly mute. Dr. Ambar Vyas is presently working as Associate Professor at University Institute of Pharmacy, Pandit Ravi Shankar Shukla University, Raipur, Chhattisgarh. Sir has completed his postdoctoral from University of Minnesota, United States in the year 2017. Sir has nearly 5 years of research and 13 years of teaching experience. Adding to his credit, Sir has more than 60 research review papers, 10 book chapters and 5 books published in national and international journals of repute. Sir holds the position of President in Chhattisgarh State APTI. With these words, I would like to request Sir to deliver his deliberation.
Thank you, Sandhya. So I'm sharing my slides. Is it visible to everybody? These slides?
Yes, sir.
Okay.
Yes, sir. Slides are visible, sir.
Can I start now?
Yes, sir. Kindly start, sir.
So, first of all, I will.
Sir, if possible, make it full screen.
Is it okay now?
No, sir.
Are you not getting full screen?
Sir, it's
No,
not
sir, no,
showing
sir.
the full screen.
Just a minute. Now is it visible?
Yes, sir.
Yes,
sir.
yes, sir. It's okay.
Thank you, Sandhya, and thank you, Dr. Rajnikant.
Welcome.
Thank to all the participants.
Rajnikant, sir, kindly mute your mic. I request you to kindly mute your mic. Yes, kindly start, sir.
Thank
Ambar,
you,
sir.
Yeah, thank you. So I'm delighted to be one of the speaker in today's first session in the figure, you can see that these autoinducers of various shapes has been prepared by Lux I oblique Lux I homolog. So it is the part in the cell body which produce these Lux I autoinducers. And when these autoinducers are produced by Lux I homolog, these autoinducers, when inside the cell, they are received by Lux R homolog. It is another receptor of these homologs, these autoinducers. And when these autoinducers get, because of their shape or particular morphology, they get fits in this homolog, and these homolog then travel and this formation or complex is known as a formation of a dimer complex. So when there is a dimer formation of dimer complex, this dimer complex goes to the Lux box. Okay, here you can see that in the figure, this complex is going to a Lux box. Oh, what is a Lux box? Lux box, it is a part of the DNA, bacterial DNA, where this dimer fits. That is Lux AHL dimer go and bound. And when it bounds, it gives signal to the cell to produce a particular type of protein. And when the bacterial cell produce this particular type of protein, they will show different reactant like inflammation in the body, virulence, any other modification in the cell. So this what we are showing, it is a single cell. Okay. So here also you can say in the figure, these autoinducers are 2 or 3 types of shape. One is capsule shape, long, one is tablet shape, and one is pentagonal shape. So those who are in round shape and pentagonal shape, they are not specific to Lux R and Lux R homolog, while AHL, that is specific to Lux R homolog, are binding to the particular site. So what does it mean? So these 3 autoinducers indicate 3 types of, we can say that, or we can compare them with the human language. So if you are not knowing the language of a person whom you are talking, if he is a Tamil speaking and you are Hindi or English speaking, then he cannot understand your language. While the capsule shape AHL, homoserine lactase, the full form is N-acyl homoserine lactose. So when this AHL is of the same homolog, then it gets fit into the receptor, and this receptor then this compound is known as Lux R-AHL dimer. And this Lux R-AHL dimer, when bound to Lux box, so what is Lux box? It is the place in the genetic DNA of the genetic material of the bacteria where it get fixed, and when it get fixed, it gives signals to the gene to produce a type of protein. And when this protein is expressed, the number of different types of reactions start in the body. So this is what its mechanism of auto-induction. So in this process of quorum sensing, bacteria communicate via secretory signal molecules which are known as autoinducer, in which contribute to regulate the expression of particular gene, what we have seen in this figure. So as I have told you, these are like language between the human beings. These signals varies between different species. So it doesn't mean that all the bacteria produce the same signals or autoinducer. It varies between species. Some bacterial species can interpret different signals, while other respond to a few types. So it is just like a person is able to communicate in 3-4 languages. So there are some species which can produce the multiple type of autoinducers, while some are specific, means there is no a few language or single language. This quorum sensing process in bacteria is regulated by various chemical signaling molecules which act like a specific language for communication, and these signaling molecules are referred as autoinducer. Now, these bacteria are known to produce signal molecules both inter and intra-species communication. So these languages are for talking within the colonies of bacteria or within the different species of bacteria. So generally, most gram-negative bacteria, okay, you are seeing that AHL. So AHL, you can say it is an signal, or you can say, or compare with it is in a language which gram-negative bacteria speaks. Or we can say that for talking with each other, gram-negative bacteria secretes acyl homoserine lactones, which are commonly known as AHLs, as their language for communication. Also, the other type of bacteria, that is gram-positive bacteria, they generally use AIP, means auto-inducing peptide, or you can say it is their language just like Tamil or Hindi or English or any other foreign language, to communicate between themselves. So these universal language used for interspecies communication is same for all the bacteria, which is called as auto-inducer 2, that is AI2. So it is a common language which all the bacteria say. Coming to the next slide. So this is what the previous figure was. You can see this previous figure. It was for gram-negative bacteria. That is schematic representation of AHL-based QS system. So this AHL means acyl homoserine electron. So this type of mechanism gram-negative bacteria. While in figure T2, you can see that it is a schematic representation of AIP-based quorum sensing system. So AIP-based quorum sensing system means auto-inducing peptide. AIP means auto-inducing peptide. Generally, this mechanism is shown by positive bacteria. Okay, here also you can say that RNA2 and RNA3, it is in series of the chemical signals which has been released by gram-positive bacteria. And when these signals are released by gram-positive bacteria through AGR-A and AGR-C, they give AIP, release of AIP, which is known as auto-inducing peptide. And when these auto-inducing peptide are released, they bound to AGR-B and SPSB, and because of that research, they give signals to the gene to release particular type of protein. So this is the mechanism of gram-positive and gram-negative bacteria. So how these quorum sensings work? So with this figure, you can see that how this quorum sensing is working. So you can see that in figure 1, when the extracellular concentration is less than the intracellular concentration. So these red dots, what we are talking about, so we are typically speaking in case of gram-negative bacteria right now. So, and these red spots refers to the auto-inducers or chemicals, or you can say that they refer to the AHL. Okay, AHL system. Okay, that is acyl homoserine electrons. So when the concentration of extracellular HAL is less than the intracellular concentration, in this case, you can see that what is the interpretation? Cell continues to replicate. Means when the concentration inside the cell is less, the bacterial cell still multiply to grow their colonies. But after some time, when the extracellular concentration become equal to intracellular concentration, you can see in the figure, then replicated cells also begin to produce auto-inducer. They used to produce auto-inducers. And when after some time, when extracellular concentration exceeds to the intracellular concentration, as it is evident in the figure, intracellular auto-inducer, which is being secreted in the cell, stops moving out of the cells. So these signals stop going out of the cells, and at the concentration, when higher auto-inducer concentration downregulates itself by inhibiting its own transcription factor, extracellular concentration again decrease in comparison to intracellular concentration, and again the first step will go. You can see that in first step, extracellular concentration is lower than the intracellular concentration. So this is how these cycles continuously grow, and the bacterial colony keeps on multiplying, and this keeps on multiplying, increase in the infection or in a particular type of the disease. So this is how it is a feedback mechanism which work through the AI, that is auto-inducer, depending upon the concentration of auto-inducer inside the cell and outside the cell. So before going to the worries related to the quorum sensing, what we are studying and why we should know that. So all of you might be familiar with the term of drug resistance. Okay, so what is the drug resistance? So as you know that recently or few months back, you have seen the advertisement in the television that because of the drug resistance, the drug is not able to treat the patients. Yeah? And before few years, there was theme of pharmacy week that don't take the medicine by yourself. You need to consult to a doctor, because as we are seeing in this growing world, people are taking medicines by themselves, and especially when you take antibiotic and doesn't complete the course, it led into the resistance, antibiotic resistance, which is known as antibiotic resistance. And this is one of the major cause nowadays. And because of this, these antibiotics are not able to treat the various disease. And this is a major problem which is highlighted by WHO. So in that case, WHO has said that to use a combination of antibiotic to overcome this drug resistance. And because of this research, you know that will also stop the drug resistance. Okay, so we all are from the pharmacy field, and we know that because of overuse of medicine or inappropriate use of medicine, especially antibiotic, the drug resistance develop. So how this does the drug resistance develop? There are 4 basic mechanisms because of which drug resistance develops. So before going to how this drug resistance develop, I would like to share you that what do you mean by drug resistance? So drug resistance means, or antibiotic resistance, especially we are talking about. So antibiotic resistance means when antibiotic which is to treat any particular infection by killing the bacteria, okay, does not work because of some reason, and unable to kill that colony of bacteria and unable to treat the disease or in the infection or control the infection, this situation is known as antibiotic resistance. So there are 4 common mechanisms by which this antibiotic resistance generally occur. The first mechanism is alteration of cell permeability. So before going to that, I would like to tell you how these bacteria work. So when you get any infection in your organ, say eye, there's an infection in the eye, or there's an infection in a wound, or there's infection because of any disease to any organ of the body. So what happens? The bacterial colony of a particular bacteria, depending upon there are 2 major class, gram negative and gram positive, they get developed in that organ. And after some time they grow and grow and grow, and in few hours, depending upon the what type of bacteria, their number increases to thousands and lakhs and millions and billions. And because of that, when they grow in a particular number, okay, so they start producing particular type of proteins, and these particular types of proteins are chemical signals which cause inflammation in the body, which cause dysfunctioning of the cell membrane, which cause dysfunctioning of the metabolism of the drug, and the disease and the drug what we are taking does not work. Okay, so this is the bacteria which forms this type of condition. Okay, so how they are protecting themselves? So when the bacteria grow in a sufficient number, they used to communicate with the other bacteria which is in the vicinity of them by releasing some signals. Okay, and this type of talking or language is known as quorum sensing, what we are studying. And these bacteria, when they multiply, they give signals that they have to increase their number, because you know that the life of the bacteria small, so they increase their number, and after a certain time there is a value called threshold value. When this threshold value reaches, these all bacteria produce a particular type of protein or glycoproteins, and these proteins or glycoproteins, they form a barrier which is known as biofilm, okay, as a covering to their colony. And because of that covering or biofilm to their colony, no drug can go inside that organ. So that they does not allow, or this biofilm does not allow to go inside that organ or that cell. And because of this protection, it is just like an umbrella. When we use our umbrella to rain, this bacterial colony use biofilm to protect themselves from the antibiotic. And because of this development of biofilm, and yeah, second thing is this biofilm development is very fast. As you know that bacterial colony grows very fast. Okay, and because of this formation of this biofilm, the drug does not able to penetrate inside the cell and kill that bacteria, and because of that we say that this antibiotic resistance or there is an infection. So these are the 4 common mechanism which cause the drug resistance. First is altering the cell permeability, which stops the influx of the antibiotics. So the bacteria, the group of the bacteria, they are highly mutant bacteria. They are able to mutate themselves very fast, just like corona virus we have seen that. So when they first time come into contact of any antibiotic, they mutate themselves, and what they do, they start the producing certain types of proteins or lipoproteins, and because of that, the composition of their wall changes, and because of the result of change in composition of walls, the permeability of the antibiotic to their cell decreases. So the antibiotic what we are taking to treat the disease does not go inside the cell, and it does not go inside the cell. It is not able to kill that bacteria. This is one of the mechanism. Second mechanism is by making the antibiotic bound cellular targets. So when antibiotic go inside the cells, there are certain targets in the bacteria to which the antibiotics bound and stop the cell synthesis, or called stop the DNA synthesis, or do any mechanism by which the death of the cell of the bacteria occurs. So what this bacteria do? This modify this antibiotic bound targets by passively modifying them. So they modify these targets passively, and because of the modification of this target, which is an easy approach, the antibiotic is not able to fit in that receptor, and once if it does not fit in that receptor, means it does not cause cell death. The third mechanism is the antibiotic is made ineffective by enzymatic cleavage. Are able to produce certain enzymes in a large amount, which called the cleavage or biotransformation of the drug before acting to the receptor. So as you know that the drug act through the receptor. So before reaching the drug, they used to release certain enzymes, and these enzymes cause the cleavage of the antibiotic. This is the third mechanism of antibiotic resistance. And the last and fourth antibiotic resistance is by controlling the efflux pumps to outset the antibiotics from the cells. So there are, as you know, that in every cell there are pumps known as efflux pumps. And what is the role of these pumps? The role of these pumps is to send out material which has been found because of the cell activity. So these are known as efflux pumps. So they control these bacteria control their efflux pumps, and if somehow the antibiotic reach inside the cell, so before reaching to the target or the receptor, they send it out by controlling this efflux pumps, and because of that the antibiotic does not work. So these are the four common, and all these mechanisms happens because of the synchronization working of this bacterial cell. And because what is the reason of the synchronized working of bacterial cell is the language in which they talk through chemical signaling, or what we say is quorum sensing. So that is why we are studying about this quorum sensing. So what are the major worries about the quorum sensings? We'll see one by one these worries of the quorum sensing. Quorum sensing led to the biofilm formation. As we have seen that biofilm has been found to affect the process of efficiencies in agriculture and aquaculture, and also in human cells. And because of formation of these biofilms, the antibiotic does not able to penetrate inside the cell, and because of which they are not able to work properly. Biofilm form on the reverse osmosis membrane used for production of drinking water wastewater recurrence, resulting direct economic loss. This is the another reason, which apart from the agriculture, it is also polluting the water. Quorum sensing based biofilm formation infectious bacteria. They protect the infectious bacteria. Quorum sensing, signaling, activation, and biofilm formation leads to the antibiotic resistance. As we have seen that it is the major reason because of which the antibiotic resistance has been caused, and the rapid deterioration of building and monuments of archaeological value where biofilms have been known. So even the archaeological value, monuments of archaeological values, biofilm formed because of different bacteria in the environment, and it led to the damage of them. Also, apart from this, quorum sensing based biofilm formation by infectious bacteria led to the antibiotic resistance. And because of all these reasons, quorum sensing need to be studied thoroughly and need to be stopped, so that an easy treatment can be given in all the sector, like it's in water, in agriculture, in disease. So these are the five major worries for quorum sensing. Coming to quorum sensing inhibition or quorum quenching. So this is the main part where the pharmacist plays its role. So till now, what we are studying about how these bacteria are talking, and what is the result of their talking, that is quorum sensing, which was a hurdle for treating any disease. So we have found out, or the people or the researcher found out, that by sensing or understanding their language, and stopping them to talk, you can say that is quorum quenching or quorum sensing. Inhibition is one of the major way has been found. Now, this bacteria from the text, from the figure, you can see that when the bacteria are able to communicate with itself, they are making in a group. Okay, and because of they can able to talk through quorum sensing, it results in formation of biofilm, and because of the formation of biofilm, the drug does not able to kill them, and because of this quorum sensing, virulence, vector expression, motility, and drug resistance, which are the common phenomena in health systems occurs. But there are certain chemical molecules. Here it is given one of the molecules. So these molecules are known as quorum sensing inhibitors. They are capable of inhibit their talking by more than 80%. And once if you use this molecule, and because of using of this molecule, the bacteria are not able to communicate with the other bacteria in the vicinity, and it get confused, or it to produce what it want to produce, and because of this loss in communication between bacteria, which can be one of the best approach to acquire the antiviral resistance strategies, or to use the drug in combination with this quorum sensing inhibitor. It can be a major way to overcome the drug resistance. So QIS could discover wide applications. So we'll see what are the wide applications of this quorum sensing. So to investigate the investigation of quorum sensing and quorum sensing related phenomena, so we need to know more about their language and the chemical signals which they use to produce mammalian resistance components against the microorganisms. So what are the ways in which the mammalian resistance occur? So and combat microbial resistance, pathogenesis, virulence, and commercially in the food industry, agriculture, aquaculture industries, power generation, paper making, and water treatments. These strategies or application as a novel antibiotic compounds can overcome the antibiotic resistance. Now coming on to the next slide, which deals with the strategies for quorum sensing inhibition. So what are the general strategies which deals with the quorum sensing inhibition? So here we can see that generally there are three strategies which are applied to stop the communication between this bacterial colony. You can see that target signaling generation. Okay, so because of this target signal generation, single precursor and signal. So we'll stop with the red cross. You can say that these signals between the signal, precursor, and signal. So there is a precursor which is released in a chemical form. If this precursor has been bind or quenched or utilized by the use of any other chemical agent which resembles to the receptor, in that case the signal will not move forward, and if the signal will not move forward, there will not be any signal to the receptor, and hence the process can be stopped. So this is known as target signal generation. The second approach is known as targeting AHL signal dissemination. In this, you can see with the help of that red cross, it has been shown that the signal precursor used to produce the signal, but the chemical signal does not reach to the signal receptor. This is the second place where we can stop the signal of chemical inducer. Again, the signal will not reach to the receptor, and again the communication will be stopped. The third is known as targeting the signal receptor. Targeting the signal receptor means we will not stop the at these 2 points. Instead, we will produce certain molecule, so which will go to the receptor and bound because of the structural homology or chemical homology. And because of this structural homology or chemical homology, our material which we are using, say it can be a plant flavonoid, and recently it has been found that there are certain plant indole derivatives which are having the same structure as that of signals or autoinducers. So because of this competition inhibition, they go to the receptor and bind the receptor, and once they will bind the receptor is not available for the process will be break. So these are the three common strategies which has been used in order to break the communication between the bacteria. So this is the slide which shows the mechanism of quorum quenching by nanocarrier. So what is this quorum quenching? Quorum quenching is a way in which we are trapping these autoinducers molecules by use of various drugs and novel drug delivery system. So once this system of language between or talking between this colony of bacteria stop, they will not be able to produce the virulent effect. Okay, so with the figure you can see that in this figure. So various quorum quenching strategies has been proposed to interrupt the cell-to-cell communications. such as removal and degradation of signaling molecule. In the last slide, we have seen inhibition of synthesis of signaling molecule, inhibition of signaling molecule receptor complex formation, and inhibition of binding of signal transducer cassette. And the nanoparticles, generally they can be, it has been also shown in this figure that this nanoparticle can exhibit the anti-quorum sensing potential by employing one or the more of these strategies to combat the drug resistance. So you can see in this figure that the red color signal, it is just like a power signal, electric signal. So this is the reported site for action of nanoparticles. So in the figure you can see that when this round brown color or reddish brown color chemical signals which are produced by synthesis molecules of these molecules, so at the first break is here. Okay, so here when these signals, if they are blocked, they will not move on to the outside of the cell molecules. Means these chemical molecules will not go out of the cell. Once they will not go out of the cell, the other bacterial cell which is in vicinity of this cell does not able to talk with this cell, so it will not able to multiply. So this is the first place where we can stop them. If it is not possible to stop them here at this inside the cell where it is produced, then again these signal molecules will travel outside, and where we can cause the degradation of the signal molecules by use by nanoparticles or the drugs. And once the degradation occur, they will change from the round shape, they will change in the different shape, and once their shape is changed, they will not able to fit in the receptor. The third target is, if it is not possible to stop here with the use of this nanoparticle or novel drug delivery system or combination of this quorum quenching agents, we will stop them from binding to the receptor. And once if they will not bind to the receptor, they are not able to send the signal for induction of quorum sensing regulated genes. So these quorum sensing regulated genes which are situated inside the cell, so they will not get the signal, and once they will not get signal, they will not express their activity. So if you are not able to stop here, then what will happen? Then in this case, these quorum sensing regulated genes which are able to produce bioluminescence. So it is good for aquatic animals, symbiotic relation, violence production, biofilm formation, pathogenesis, lymphocyte synthesis, and swarming motility. These are all are the negative effect of the bacteria which it causes when the cycles get over. And once if you stop this cycle, the bacteria is not able to communicate itself. Or again, the last is the expression of quorum sensing regulated genes. If somehow the genes get the signal, but if with the use of this nanoparticle or quorum quenching agents, this expression can also be stopped. So there are 1, 2, 3, and 4 sites at which we can stop this quorum sensing activity by use of quorum quenching chemical agents through the novel drug delivery system, which are side-specific, just like nanoparticles. We will also see the other delivery system. There are, apart from this nanoparticle, there are another novel drug delivery system which are used as a promising strategy to quantum the quorum sensing in case of infectious disease, which are under the research, and few of them are still there in the polymeric nanoparticles, polymeric microparticles, hydrogel nanoparticles, lipid carriers like liposomes, solid lipid nanoparticle, phospholipid micelles, and metal or inorganic nanocarriers like gold nanoparticle, nanoshell, magnetic microparticles, nanoparticle which are not mentioned here, and quantum dots. So these are different types of nano drug delivery systems which are used for eradication of bacterial resistance by the encapsulation of quorum quenching inhibitors, and because of which the quorum sensing and biofilm formation can be stopped. There are few certain examples of the research has been done by various scientists, and they have shown that successfully that it is able to stop this process of bacterial communication. So the first example is Ketosyn polymer nanoparticles using zinc oxide, and here it is a flavonoid eugenol has been used, and it explores the benefits of applying Ketosyn in endodontics. And the polymer which is used is Ketosyn, and it causes enterogenous fasciitis pathogen, is a pathogen. So with the use of this eugenol, this communication has been stopped. Apart from this, gentamicin, amoxicillin, another antibiotic used nanoparticles and lipid nanoparticles, so increasing half-life of the drug, enhancement of efficiency of Precautions and biofilm forms, and the polymers apart from Ketosyn which has been used is PLGA, that is polylactic chloroglycolic acid, and pectin sulfate mixed with lipid, and these are used for different types of bacteria, Pseudomonas aeruginosa and Halobacter pylori, which are found in stomach pylori, and aeruginosa is for ocular infection, one of the bacteria which cause major ocular infection. Also, other bacteria which has been stopped for quorum sensing, that is Staphylococcus epidermis, with the use of different types of nanoparticles or novel bacteria done. Again, another slides which shows these examples. Here there are a number of examples. I'm not going to read out every examples. So I have used 3 slides of this novel drug delivery system. So containing lipid nanoparticle, containing metal-based nanoparticle, containing polymer-based nanoparticle. It is just to show that a large number of research has been already done, because these studies are found very successful. And soon there are 2 or 3 patents has already been done, and 1 or 2 products are there in the market. So because of which these products are gaining more popularity, and there is an ample scope to use these quorum quenching inhibitors as a novel drug delivery system. So that is why I'm more to use 3 slides to show that how the number of different novel drug delivery systems are used for overcoming the quorum sensing or by use of quorum quenching inhibitors. This is another example. Now we can see that there are various strategies how you can, in this slide you can see that there are different nanoparticles, how these nanoparticles they are working working in combination with quorum quenching inhibitors. They have certain advantage and there are certain limitation also which has been discussed here. So there are 2 slides for them. We'll see one by one. So the first example is silver and other heavy metal nanoparticles. So these heavy nanoparticles or metal nanoparticles, they have advantage of killing the major gram-positive and gram-negative bacteria. They can kill both type of the bacteria. They are easy to synthesize because of physical, chemical, and biological methods. Apart from that, there are diverse formulation possible coating or emulsion because they have a metallic core. So coating or emulsifying them is very easy, and obviously because of their metallic nature, they are able to cross the biofilm also. If some level of biofilm has been it has been proved that this metal nanoparticle, and they call synergistic effect with antibiotic. But the limitation with them is the mechanism of biofilm penetration and killing is not fully understood till now. And also there are certain metals which are toxic, toxic effect which need to be studied before launching this as a successful drug delivery system. Gold nanoparticles, they are easy to synthesize. Like other nanoparticles, metallic nanoparticles, they are easy to tunable, change their shapes, and they have good photothermal property, but the killing is only demonstrated on limited number of strands. So these gold nanoparticles are not widely expected for the strands. So they have one limited number of strands. Again, they have potential cytotoxic, storage stability, and above all, they are very costly. Apart from that, carbon-based, graphene-based, and polymer nanoparticles, which are easy to tunable, and they have good stability also, but they are easy to tunable. And still a large of research has been going in polymeric nanoparticle because they have potential advantage of mucoadhesion, especially to the organs like ocular eyes. Another mesoporous silica nanoparticles, liposomes, and polymer-based nanoparticles, but specific target specificity, and they are more specific to kill this bacteria and stop this quorum sensing. This is another example. So the problem with the available drugs, so reaching to the conclusion. So the problem with all the available drug delivery system is they do not give drug dual response. So all the antibiotics, or especially we talk about the drugs which are used for ocular or other type of infection in the body, they are not able to give the dual responses. They give only one response. But when you are using both quorum inhibiting agent, which are generally plant flavonoids, and recently there are 12 indoles which are derived from plants has been to stop or inhibit this organism, and of biofilms in combination with two strategies will use to cure, so it will be helpful to cure the infection, and they will also result in reduce the dose dependent side effect of the antibiotic. So once the dose of antibiotic is reduced, obviously the drug resistance to antibiotic will be reduced, and therefore there is a need of developing such drug delivery system with due combination of the antibiotic as well as quorum sensing inhibiting agent to overcome these problems of drug resistance. Thank you so much for your patient hearing. And before thanking the management and the organizer and the principal of the college, I would like to ask from the students if they have any questions, or the participants if they want anything to share, or something which I was not able to clear them. So the solution for their doubts now. Thank you. Anybody? Does anybody has any question?
Participants may ask any question if they have. Any participant?
Willing to ask, there is on the first slide, you can have my email, Vyas@gmail.com. So if anybody has any query and they want to start right now, so I would like to share with you that one of our research scholars was RGNF, Rajiv Gandhi National, working of this type of work. So one of our research scholars in University Institute of Pharmacy, Pandit Ravi Shankar University, is being working on this mechanism on ocular infection of gram-negative bacteria. So we are using this approach, how much we are able to lead in this way. So that was my interest to discuss this topic in today's conference. So we are already working. So any of the students who have any doubt or want to practically work on this can contact me. Thank you, Sandhya Mishra madam. You can proceed.
Yes, sir. Thank you, sir, for sharing your profound knowledge with us, sir. Sir, we are highly obliged to have you as our speaker for today's session, sir. Thank you so much, sir.
Thank you so much, sir.
Thank you, Dr. Rajnikant. And again, I would like to thank management, Columbia Institute of Pharmacy, Senior Professor of the Institute, Dr. Ravindra Pandey, sir, Dr. Shirivastava Shukla, sir, and organizer or convener of this DBT sponsored Rajnikant connection.
Thank you, sir.
Thank you, sir. Thank you so much, sir.
Students, hope I have finished. Thank you to all of you.
Thank you so much, sir. Sandhya ma'am, next speaker has joined Dr. Rishi Paliwal.
Yes, sir. Good morning, sir.
Good morning,
Paliwal,
sir.
Now we have with us Dr. Rishi. Good morning, sir. Now we have with us Dr. Rishi Paliwal as our second speaker. I welcome Dr. Rishi Paliwal on behalf of whole Columbia Institute of Pharmacy Raipur team, sir. Welcome, sir.
Thank you.
May I now take this proud privilege to introduce our speaker to the participant? May I, sir?
Yes, please continue.
Dr. Rishi Paliwal is presently holds the position of Assistant Professor, Department of Pharmacy, IGNTU, Amarkantak, MP. Dr. Paliwal has completed his postdoctoral from Rangel College of Pharmacy, Texas, A&M University, USA. Sir has 14 years of teaching and research experience in the field of pharmaceutical sciences. Sir has published 40 research papers in high impact factor journals. Sir has also contributed 18 book chapters in the leading international books. Adding to his credit, Dr. Paliwal is a recipient of Rudolph Simedin's Award, Career Award for Young Teacher, and Outstanding Pharmacy Teacher by APTI MP branch. With these words, I would like to request Sir to deliver his deliberation. Please start, sir.
All right. Am I audible?
Yes, sir. You're audible.
audible.
All right. Thank you so much, Rajnikant, Dr. Rajnikant, and all the team, Dr. Amit Roy sir, and all my dear colleagues over there for inviting me for this conference. Thank you. And I think that I will try to my level best to discuss some aspect that I could understand in this area as per my experience. Before starting, I would like to know that if everything is okay from my side, my voice, and there is no echo probably. And
Yes, sir. Your
I
voice
think
is
that
clear, sir.
Dr. Amar Vyas sir, नमस्कार सर, कैसे हैं आप?
मैं बढ़िया हूँ ऋषि सर, अच्छा लगा आपको देख के और मैंने 8-10 मिनट आपका ले लिया एक्स्ट्रा, काइंडली एक्सक्यूज मी फॉर।
नहीं सर, कोई इशू नहीं है सर। फर्स्ट ऑफ ऑल तो आपको बधाइयाँ सर एसोसिएट प्रोफेसर बनने की, और हम लोग जल्दी मिलेंगे फिजिकली। थोड़ा सा बिजी था, इसलिए थोड़ा आपका लेक्चर ज्वाइन नहीं कर पाया, एग्जाम कर पाया।
कोई दिक्कत नहीं सर, आप आइए, मैं हूँ आपके साथ।
सर so Amber sir, if you allow me, you please stop presentation so that I can start my presentation.
Okay.
Sir.
Hope it is done now.
Okay, thank you so much. Thank you. So, just a minute. Going to start. Just a minute. And then I think it will be best when I'm painting a window. Okay, that's right. So is my slide visible now? Please confirm.
Yes, sir. It's visible, sir.
It's visible.
All right. Thank you so much. If there is any disturbance, you can please let me know. I understand that in technical regions there may be some problem. All right. So
Sir,
thank you so much, everyone. I'm here.
Sir, I request you to kindly display the slides in full screen, sir.
No, it is
possible,
in full
sir.
screen mode actually.
Okay, sir. It's not showing.
No, it is in full
here,
screen mode.
Okay, sir.
Just a minute. It is in full screen. Is it okay now? No? Okay.
No, sir. It's not showing over here full screen.
I'm changing different mode. Okay, let me just wait.
Okay, sir.
Okay, so I'm going to not entire screen. It is create an issue. Entire screen. I think this is not. Is it okay?
Yes, sir. Yes, sir. It's full screen, sir.
All right. Thank you so much. So, good morning, everyone. This is Dr. Rishi Paliwal from Indira Gandhi National Tribal University, Amarakantak. And as the topic is there, I am going to discuss something which is about green synthesis of metallic nanoparticle, especially used for diagnosis and therapy of different infectious diseases. All right. So, in background, this is the picture of my university. You can see the green environment. It is a very good environment here, and we are living with the surrounding of the plants. We see that the plants are most since ancient time, and we are surrounded by them, and we are having a lot of ecological system, and different variety of plants are there. And they are very good friends. They are giving us oxygen and other required material for living. So how these plants are, definitely they are part of ecosystem. So the approach which is using any plant and not the solvent may be a more eco-friendly and more environmental friendly approach. So here in this presentation, I'd like to discuss you these plants or other biological material systems that we use for synthesis of nanoparticle nature, and how these metallic nanoparticles are used in infectious diseases. So no need to describe that. We know a lot of plants. They are all around us, right from ginger, cumin, aloe vera, nutraceutical, pharmaceutical, immunomodulatory properties, but also in our characteristics, which are very important, flavoring agents, etc. So using plant in every part of it for every day-to-day activities, and as well as the well-being, different aspect of being. Okay. They have different type of pharmacological activity. We know that they have anti-inflammatory activity, antioxidant activity, anti-cancer, cardiovascular, neuroprotective, and many more, including antimicrobial properties. Compounds from these plants which already act as an antimicrobial agent. But aspect of these plants can be used at different part of the plant for making nanoparticle that we are going to discuss. We know that they are working as a different, in case of cancer, for example, they are working as a free radical scavenging agent. A lot of phytoconstants are there, and they are इस तरीके से they are working in our direction where we can live better and we can prevent many diseases. So in prevention as well as they are very effective in different types of vitamins are there.
Hello.
So what we do is we prepare different type of formulations from the plant. We use them. Yes. Is there any problem? Somebody was saying something. Is there any issue?
Javendra Kumar.
Everyone, please put your mic off. If you have any question, we will discuss in the last. Okay, no problem. All right. Okay. Should I continue? So these are the different aspects of the plant. We usually make different type of formulation from these plants, and in general, and we are using them from many ancient times. As the evolution of nanotechnology took place, and we are looking for the different aspect of nanotechnology, as today we are discussing their application in infectious diseases. So atom nanomedicines came in this in the existence, and it is the application for either therapeutic or diagnostic or sometimes both. And we are using the very nano scale level and their properties, which is not possible through the micro or macro systems. So you can see these are the different type of range of nanoparticle which can be used for different purpose, and they have very minute size, right from 1 nanometer to 1000 nanometer, we can say in detail. For that, we need different type of material. We can prepare polymeric nanoparticle, we can prepare lipid-based nanoparticle, we can prepare metallic nanoparticle. So for development of any nanomedicine, these three important aspects are there: material, process, and regulatory aspect. So we have to take consideration that which particular material is more useful for our case, like in case of our choice, if we are going for a delivery of any anti-infective agent, we can use a polymeric or lipid nanoparticle. But we want some metallic nanoparticle as a tool for anti-infective agent, antimicrobial, antifungal. So in that case, we need the metallic nanoparticle preparation. So there are several aspects that we have to consider in preparation of nanoparticle in particular. So the question is whether our prepared preparation would go reach to the market or not. For that, you have to understand different components, which are maybe material component, maybe process component, maybe interaction of ingredient with each other, and the performance of the final nanoparticle, size, shape, charge, and many properties. Whether the prepared nanoparticle could be sterilized if it is for parental prepared. The components are acceptable by the regulator at the large. And what about the characteristics of these nanoparticles, whether they will be retained in animal preclinical studies or not? And then what will be the stability of these nanoparticles? ये stable रहेंगे कि नहीं रहेंगे? Same size और charge उनके ऊपर रहेगा after storage time, and reproducibility of the procedure. Every time the same size and same shape can be achieved or not. अगर हम large scale production करेंगे, then what will be the method and how industry will adopt it? So different type of nanoparticles are prepared by different methods. For example, if you are dealing organic nanoparticle, maybe polymeric or lipid, so we use the different techniques. But in case of metallic inorganic nanoparticle, which are metal, we most use the physical technique or chemical technique for different type of nanoparticle. Different techniques that are reported in the literature. But friends, I would like to say you one thing. There is one very common question that sir, which method is best for developing nanoparticle? There is no universal method for developing nanomedicine. Depends on the what material you are in, what type of purpose is there, what type of characteristics you want in nanoparticle. Accordingly, you choose the different type of material or process, both. So you can see here I have given some highlight of some polymeric nanoparticle, different methods and their limitations and remarks, and similarly with the lipid nanoparticle and so on. So before making and discussing about the importance of nanoparticle, I just want to know that they have some challenges with the nanomedicines, which require the ability to develop a structure which has very precise level, atomic precision level is required in these nano devices. So shape and size, biocompatibility, powering, and communication. These four are important points which are required for successful action of these nanoparticles, whether it is controlled delivery, targeting, imaging, or anything. These four are the basics of any nanomedicine and nanoparticle. We know that sometimes if we are loading some drug into that for anti-infective agent, antimicrobial activity, some antifungal drug is loaded on some nanoparticle, for example, then reproducible drug release profile is also one of the requirement, and batch to batch uniformity is also one of the requirement, so that we can establish a quality IVC performance in our nano drug delivery system. And after crossing the several barriers in the preclinical level, whether there is a therapeutic need, whether there is a market demand, whether there is a research and development, whether production steps are sufficient, scale of feasibility are there or not, clinical trials are possible or not, and what about the regulatory issues. इतनी सारी चीजों के बाद one nanoparticle or one nanomedicine product reaches into the market. अगर उसका market demand नहीं है, तो then there is no use of that nanoparticle, and it will be very costly, and industry will not be ready to prepare. So these all are the challenges how nanomedicine is developed, and also the grass status of the material, because it should be safe, it not be, and toxicity, especially with the help of size and shape of the nanoparticle, and what will be the biofate of these nanoparticle after administration. Body में जाने के बाद वो degrade होंगे नहीं होंगे, कहाँ deposit हो जाएंगे, toxicity cause करेंगे क्या, and balancing between the multi-component system at large scale production. जब आप high level पर आप बनाते हैं, तो जो different component system है वो perfectly balanced होने चाहिए, otherwise market नहीं कर पाएंगे, हम उनको हमारे features बदल जाएंगे. So these are some examples. I am not going in detail. I am coming to the topic now because we are developing the nanoparticle through the green chemistry. So I just would like to highlight what is chemistry in general actually. So we know that green chemistry means, as you know, the green means environmental friendly, and there should be no use of organic solvent somehow, and we are using the perfect balance of this chemistry so that we can develop the more eco-friendly and sustainable product. So if we talk about the different controls, safety control of green chemistry, so when you are dealing with nanoparticle or in any kind of chemical reaction, we know that personal protective wear, and everyone has to wear these things in the lab, and especially with the organic solvent, there are more chances of hazardous reactions and actions whereas in plant there is no issues as such. Better engineering controls are there, and it eliminates the exposure of the organic solvent to the one who is working with the development of nanoparticle. So these are the different pillars of green chemistry, and this concept is for reduced toxicity and energy efficiency, better use of biological materials and better quality control, waste reduction. All these atomic economy की तरफ हम बात करते हैं, that is the key feature of the green chemistry. So होता क्या है, हम करते हैं green chemistry से lab में, you can use the and you can extract these plant for different purpose, and organic solvent, especially water, and so no water as such, we can also get the phytoconstituents by degradation. So we can get the phytoconstituents in our extract or in our distillate, and that we use for the development of nanoparticle. If you are going to make the nanoparticle like gold, silver, palladium, etc., then there are three different methods: physical, chemical, and biological. So physical method required high energy, chemical method required the chemicals and organic solvent, whereas the biological methods, particularly or microorganism, any other material which is biological source, that can use to develop the nanoparticle. So we are focusing on these methods. So how the nanoparticles are synthesized in general? So if we talk about the any nanoparticle preparation method, तो आप माइक्रो से नैनो पार्टिकल कैसे बनाते हैं? देयर आर टू अप्रोच, यू गो थ्रू द टॉप डाउन अप्रोच और बॉटम अप अप्रोच. टॉप डाउन में क्या करते हैं आप एक मैक्रो पार्टिकल है, आप उसमें एनर्जी अप्लाई करते हैं, उसको तोड़ते हैं फोर्सफुली और छोटे पार्टिकल्स में लेकर आते हैं. बॉटम अप्रोच में आप क्या करते हैं? आप उस मटेरियल को डिसॉल्व कर देते हैं, मेबी वाटर मेबी ऑर्गेनिक, और जब वो डिसॉल्व हो जाता है, मोल में चला जाता है, तब आप उसको कंट्रोल्ड वे में रिड्यूस करते हैं. सक्सेशन प्रोसेस के थ्रू आप नैनो रिड्यूस होता है और मॉलिक्यूलर फॉर्म से वो आपस में प्रेसिपिटेट लगते हैं फॉर्म में, और हम उसको कंट्रोल्ड प्रेसिपिटेट दे सकते हैं, कंट्रोल कह सकते हैं, और हम उससे बनाते हैं पार्ट. ये दो अप्रोचेस हैं जिनमें हम जा सकते हैं. तो बायोलॉजिकल मेथड्स जो है वो बॉटम अप अप्रोच को यूज करते हैं, जहां फाइटोकॉन्स्टेंट्स यूज किए जाते हैं एज ए रिड्यूसिंग एजेंट फॉर any kind of metallic nanoparticle. So like already physical approaches, chemical में chemical reduction कर सकते हैं, hydrothermal purification, sol gel, polyol process, some approaches. Physical approaches में gamma radiation sonic method है, micro radiation, thermolysis, laser ablation, sputring, ये सब आप यूज कर सकते हैं. ये सब कन्वेंशनल है, हाई कॉस्ट होता है इसमें इन्वॉल्व, जब इनको कॉम्प्लेक्स प्रोसीजर इन्वॉल्व होता है. बट अगर आप बायोलॉजिकल अप्रोचेस की बात करते हैं, जहां पर हम प्लांट यूज करते हैं, लीफ, रूट, बार्क, फ्रूट ऑफ एनी प्लांट, एनीथिंग व्हिच इज अवेलेबल इन आवर सराउंडिंग एज ए वेस्ट मटेरियल, एनी बैक्टीरिया, एनी फंगाई, एनी एल्गी, एनी plant waste or living cell or tissue or organism, etc. So they are very cheap procedure. Alternative methods, very low cost, biocompatible methods, simple and eco-friendly procedure. So these are the advantages of these approaches. So अब करते कैसे हैं, बनाएंगे कैसे हम ग्रीन केमिस्ट्री से नैनो पार्टिकल? आप कोई भी प्लांट आपकी सराउंडिंग में अगर है, आपको लगता है कि उसमें फाइटोकॉन्स्टेंट्स हैं जो सफिशिएंट एबिलिटी रखते हैं, आप उस प्लांट के किसी भी पार्ट को ले सकते हैं, उसको पाउडर फॉर्म में कन्वर्ट करेंगे, एंड देन यू विल बॉयल इट एट 160 डिग्री सेल्सियस इन वाटर. बॉइलिंग करेंगे. बॉइलिंग करने के बाद प्लांट में जितने फाइटोकॉन्स्टेंट्स हैं जो वाटर में आ सकते हैं, वो सारे के सारे वाटर में आएंगे, एंड देन यू विल फिल्टर इट, एंड यू विल यूज दिस फिल्ट्रेट फॉर ग्रीन नैनो पार्टिकल मेथड प्रिपरेशन. तो उस फिल्ट्रेट में अगर आप अपना सपोज आप सिल्वर नाइट्रेट का सॉल्यूशन डाल देंगे और सिल्वर के नैनो पार्टिकल बनाना चाहते हैं, तो सिल्वर के नैनो पार्टिकल आपको मिलेंगे, या आप गोल्ड नैनो पार्टिकल बनाना चाहते हैं तो उसका प्रीकर्सर सॉल्यूशन उसमें ऐड करेंगे. ये प्लांट एक्सट्रैक्ट है, आपकी कोई भी माइक्रो ऑर्गेनिज्म का एक्सट्रैक्ट भी हो सकता है, उसको भी आप यूज कर सकते हैं. और सारे एक्सट्रैक्ट में डिफरेंट टाइप के कंपाउंड like alkaloid, terpenoids, phenolics, hemoprotein, vitamins, sugar, coenzymes, anthraquinones, etc. So they serve as a reducing agent for these silver nitrate or gold solution, and then they convert them into the proper metallic nanoparticle. Use plant मैं biological methods की बात करूँ in general, so plant, synthetic biology या microorganism. You can use any approach for making a gold nanoparticle or any metallic nanoparticle, and then these nanoparticles could be used for different purpose, like targeted drug delivery or imaging or antimicrobial or metabolic compartmentalization or anything. Okay, so this is how it is used. As I already told you, you can use suppose अगर आपको metal ionic source का plant extract के साथ reaction किया, nucleation होगा, and then there will be then kept nanoparticle बनेंगे. Some factors are there that may affect on the yield of the nanoparticle. For example, pH, maybe the concentration, maybe the reaction time, maybe the reaction temperature. So one has to optimize which one is the best condition to develop the nanoparticle. How these nanoparticles are prepared mechanistically? Phytoreductants जो होते हैं वो metal ion के साथ react करते हैं और उनके ऊपर capping agent की तरह काम करते हैं. Metal ion are captured over the phytoconstituents, and afterwards हम उनको जो nanoparticle बने उनको characterize लेते हैं through DBT and other techniques. And afterwards, if they are suitable, we can use for biomedical or industrial applications. मैं यहाँ पर आपको एग्जाम्पल लिटरेचर में पेपर का यहाँ देख सकते हैं. फ्रेश लीव ऑफ प्लांट यूज किया, उनको कट किया, ड्राई किया, वाटर में एक्सट्रैक्ट पेपर एंड देन फिल्टर किया. फिल्टर करने के बाद प्रीकर्सर सॉल्यूशन तैयार किया, प्रीकर्सर सॉल्यूशन को मिक्स किया एक्टिव सेट के साथ एंड बायोरिडक्शन होने दिया कंटीन्यूअस स्टरिंग के साथ. For some time there was the formation of nanoparticle, and then these nanoparticles were filtered and collected and then characterized. केवल प्लांट में केवल लीफ की बात नहीं है. You can any phytochemical. Here in another paper I used the theramine, इमली का इस्तेमाल किया. इमली के पल्प को लिया, पल्प निकाला और उसमें nanoparticle बनाने के लिए प्रीकर्सर को ऐड किया, उसको हीट किया और afterwards they find the good nanoparticle. Not only that, but this is the anode where the amla used umbilical fruit, and in which the extract was taken and silver solution was prepared, and then this silver solution reacted with the plant extract and prepared silver nanoparticle. Color changes and conformation was, and here you can see they have tested it against the bacteria, and they have tested against anti-infective property of the silver nanoparticle. Not only plant source, animal source. I already told you in an example, the squirrel egg was used. Yolk was taken and 99 ml distilled water was taken, and this was used as a reductant, and gold nanoparticle was prepared using this characterized. Now the question is कि nanoparticle जब बनते हैं डिफरेंट शेप, रेगुलर शेप नहीं बनता. आप अलग अलग से बनाएंगे, अलग अलग माइक्रोन से बनाएंगे, अलग अलग शेप एंड साइज होते हैं. So there are different types of particles being synthesized in nanoparticle in different such paper. This is your question. So plant extract or microbial extract will serve as a capping agent over the metal ions. Sometimes the nanoparticles are uncapped. Some of the nanoparticles are partially capped. Some of them are fully capped. So therefore, the polydispersity in the preparation and also in the different size are obtained in the nanoparticle. There is reported for plenty of green synthesized nanoparticle, and was one activity was done using these nanoparticles, and on the basis of screening of different plants, and they have found that green nanoparticles are around 17 of these antiplasmatic activities, and they were safe in the less toxic activity was obtained in case of. So these are having very small size. Metallic nanoparticles are having very small size than polymeric from 10 nanometer to 100 nanometer. Reports are used, are continuously plant and microorganism for synthesis of metal. You can see in India, one of the leading countries is this exercise and type of metallic particle using different plant parts. This is one study where something is compared that nanoparticle different plant extracts से आप बनाते हैं तो उनके different आपको sizes मिलते हैं, mix होते हैं और उनके shapes भी different होते हैं, spherical to cubic to regular shape, then oval and square shape. So it depends on, as I already told you, that depends on how metallic ions are attracting with the fight constants accordingly the size and shape. And you can see the color changes also, which is clearly peaks. So yeah, color and UV will be different depending upon the phyto constant present. So what is the evidence of this green synthesis? Rapid, cheap, and very having a scale-up technology. Synthesis procedure requirement of stabilizer or capping agent that are they are pharmacologically active gold particle which is attributed to the phytochemical biomolecule surface during this biosynthesis procedure, and they have ease of functionalization. आसानी से आप उनको further आप ligand बनाना चाहते हैं, कुछ और करना है, then you can do that, which help in decorating them with the coupling agent, protein, nucleic acid, antibody, further drug molecule. And biologically active gold nanoparticle monodispersed, highly stable to the chemically produced nanoparticle. But there are certain limitations, like for example, don't know which exact chemical component is present in plant extract or microorganism, so that we cannot that this particular plant extract is responsible for bioreduction. अगर आप एक्सेसिव या लार्ज स्केल बायोसिंथेसिस करेंगे तो इकोलॉजिकल इम्बैलेंस हो जाएगा. आप पता चला सारे प्लांट की पत्तियां आपने तोड़ डाली, लार्ज स्केल प्रोडक्शन करना है बहुत सारा, तो आपको एक रेशनल बैलेंस बनाना पड़ेगा, आपको यील्ड की तरफ जाना पड़ेगा, हाई यील्ड डेवलप करने पड़ेंगे. सारे के सारे मेटल नैनो पार्टिकल आसानी से प्लांट सिंथेसिस या ग्रीन सिंथेसिस से नहीं बनाए जा सकते, तो उनके लिए आपको फिजिकल या केमिकल अप्रोचेस यूज करने पड़ेंगे. स्टैंडर्ड मेटल साल्ट्स आप रिडक्शन प्रोसेस में जनरली यूज करते हैं, बट उनकी प्रोसेस को कंट्रोल करना डिफिकल्ट है. प्लांट में चूंकि सीजनल चेंज होता है तो कंसंट्रेशन ऑफ एक्टिव बायोमोलिक्यूल चेंजेस विद द टाइम, एंड दैट्स व्हाई द ओवरऑल एक्टिविटी इज चेंज. Now I want to share some of our laboratory experience, our own experience. We tried this in our lab. This is also known as black turmeric, which is easily available in this region, and this belongs to relatively. The colors are added into the dried powder, then the extract of this particular rhizome, and then we incubate the selected solution with this plant extract at our temperature. And we don't want to involve any heating condition. We just want to see what will happen with the room temperature, whether it is possible to prepare the nanoparticle. And we found, yes, the color changes with the different composition of the plant extract. We can find the silver nanoparticle when the dark color is obtained. This is the confirmation of preparation of silver nanoparticle. For further confirmation, we did the UV spectroscopy, where we found the peak, which is at very close peak, 442, 443, which was very reported in the literature and was close to the reported in the literature, and confirmation of nanoparticle preparation was done. I just want to highlight you one thing, that sometimes when you are doing the green chemistry with the plant, please identify the plant source very carefully, because it may be a wrong prediction, and when you are going for publication or you are going to report, it may be a different conditions. For example, in one case, I just want to show you. In one case, the authors use the rabi leaf extract for the preparation of this plant, and sometimes when you later on they somebody suggested them that this plant is not available in your region, then they see, okay, there is the correction. Okay, this plant was not, it was Moringa oleifera. So some identification of correct plant is very important, or microorganisms or green synthesis or green chemistry. So be very careful, because these plants very little difference between leaf size and different aspect of pharmacognostic features. So please be very careful. Now let us come to the next part. Suppose if we prepare the metal particle, what we will do with them and how they are effective as a antibacterial agent. So metal nanoparticle, metal sulfide nanoparticle, metal organic frameworks, metal oxide nanoparticle, top metal oxide nanoparticle. These are the different types of metal-based nanoparticle and easily prepared with chemistry. So we can these nanoparticles serve as nanopeptides. They may be antifungal, antibacterial, so they have the very strong advantage over the common disinfectant. For example, very low dose is required, 200 ppm only. Very short time exposure is required to serve as an anti-infective agent. They have high stability, they have low toxicity and unique antimicrobial properties. So because of that, main particle could be used for anti-infective agent, and I will show you how they work. So metallic particle serve as an anti-infective agent through different mechanisms. For example, some of time they work on destroyed microbial cell wall. Sometimes they develop the oxygen species and then therefore lead to. Sometimes they damage protein membranes, these proteins, and sometimes they damage by these different mechanisms. Great advantage of some particles, especially the gold one, will much explode in the system. So after preparing gold particle from green synthesis method, one can derive the particle, nanoscale, nanostar, nanosphere, nanoclusters of different size, and also functionalize further like charge, negative charge, or can make them polyethylene glycol can be on the surface. They can with DNA, or proteins, or for further diagnosis can with nucleotides or drug or gene or any probe or any antibody for selective targeting. So after making the green nanoparticle, these nanoparticles from one can body the surface. For example, this is an example where gold nanoparticle are used together for photodynamic therapy, and where the silver nanoparticle is used for killing purpose, and gold nanoparticle through NIR radiation is used for diagnosis purpose. So together they could be also used in combination. In this example, silver nanoparticle in bound healing process, they are used as an hydrogel, and in silver nanoparticle were prepared and they will was incorporated into hydrogel, and that hydrogel was applied on the surface of any bound for bound biofilm formation, and the bacteria are killed by these silver nanoparticle, and rapid bound healing activity was obtained. Similarly, report I have observed, which was on silver nanoparticle in coronavirus, SARS-CoV-2, and they have used the silver nanoparticle as an antiviral agent against the coronavirus, and they have been very proved effective as an antibiotic. For diagnosis purpose or for locations, we know that different type of microorganisms live in different parts of the cells. Intercellular locations like nucleus and even the endoplasmic reticulum. You can see in the picture some of this. So these particles, silver nanoparticle, gold, platinum, selenium, they can also reach into the intercellular location easily, and where they can show the microbial activity, and they can clearly kill these microorganisms by reaching intracellular locations, and then they kill them. So this is how by the mechanism. So this is how they are very, very effective carrier system, metallic nanoparticle for the anti-infective agents and then for treatment of infectious diseases. They can also be kept with the radionuclide on the surface or any optic contrast agent or any polymer for the like liposome, polymer, and dimer, and their forward surface be modified, and they can serve as an for therapy. So this is how they are different diagnostic and therapeutic purpose. So this is my research team, which is actively involved kind of research. We are doing activities at green chemistry, and we are utilizing plant resources for development of different particle for getting results, for especially for wound healing we have done, and we are also working in the different aspect of these nanoparticles, diagnostic particularly. So this was all from my side. I request you all to visit once. It is very beautiful place. Should be query related to my position later on as well, and email me on this address, WhatsApp me on this email, this my mobile. So this is from my side. Thank you so much for listening carefully. If you have, I will just clarify your new ideas. So this was all. Thank you very much. Hello. Hello, am I audible? Hello. If you have any questions or any suggestions, both are anything.
Sir, I think there is a network issue. We are unable to hear your voice clearly, sir.
All right. Is there any question? This is what I am asking. Any question?
Okay. Thank you, sir.
You can type a question.
Hello.
Why is not there? Yes.
Hello. Good
Yes.
afternoon, sir.
Yes.
Sir, I have one doubt about the green Govt with the green chemistry, because we know that all the plant extracts are basically phenolic and alcoholic in nature. Basically, the most of the chemical constant is not water soluble. So, is this the process is used completely go for the green chemistry? Because most of the polymers are also soluble in the alcohol, methanol, and this. So we restricted to use of the water soluble polymers for the stability.
Yes. I can tell you one thing: metallic nanoparticle. Okay.
Okay.
Where
Okay,
the gold, silver, zinc oxide, iron, metal oxide nanoparticle, if you are preparing, for that only we use the green chemistry. So these metals are easily reduced and they are converted into the nanoparticle through these plant extract. And in this aqueous extract, most of the phytoconstants that are present, maybe alkaloid, maybe phenolics, they are capable to reduce these ions from the solutions to nanoparticle. So we don't use them for polymeric or lipid nanoparticle.
Okay,
We
sir.
use green chemistry only for metallic nanoparticle, gold, silver, iron, zinc oxide, titanium oxide, like this. Metal and metallic oxide nanoparticle. Because these are very selective probe for any optical imaging. So metallic nanoparticle are very, very good for optical imaging purpose, diagnostic purpose. And also they have anti-infective properties as well, because their surface is bioactive. So they clearly reaches to the intercellular locations, जहां पर बैक्टीरिया वायरसेस लोकेट हो जाते हैं हमारी बॉडी में, हमारी सेल्स के अंदर, वो वहां तक आसानी से जाकर उसको किल कर सकते हैं. So metallic nanoparticle, I am focusing, I am not talking about any liposome or polymeric nanoparticle. Okay.
Okay, sir. Thank you, sir.
All right. Thank you. Okay. So any other question related to this? If no question, then I will, or you learn nothing. Sir, Rajnikant, can you hear me?
Yes, sir, I am here, sir.
It seems audience have no
Thank you
question.
so much, sir. Thank you. Okay, sir. It's okay, sir. Thank you so much for joining us, sir, for giving your valuable time. Because many times I have called you, sir, but you were on the right time to join us for the second scientific session. That's
No,
very.
thank you so much.
This is not for. It is a unforgettable moment, sir. Because session is already. It was getting late from yesterday. That's unforgettable.
Thank you so much. Thank you so much for inviting me, because I know that there are different lectures and schedule is going on and you have to conclude in the evening. So I want to keep my presentation only 45 minutes and 15 minutes for discussion, because sometimes students or audience do not ask the questions because of the restriction of the time. So I kept 15 minutes for that. If there is any doubt related to the method, operation, uses and applications, I will be happy to answer.
Yes, sir. And sir, your personality, just like for the physical conference, offline conference, because this is the online platform, the participants are not much strength. So very soon we are planning to organize offline conference, sir. And once again, you.
It is missing actually, because nobody. This is a monotonous side of. I am talking. No, I can't see any faces, and I can't observe where they are having questions and problems. So this is one way only. So, however, I will appreciate to the audience, if they have any doubt, they can email me. They can WhatsApp me. Otherwise, in email or offline platform, we will be here. It is a very interesting topic actually. So I will advise those who are working in the direction in planning. You can plan into the direction. These having very potential anti-infective agents in the bodies.
Thank you.
Okay,
Thank you so much, sir.
much. Thank you so much. Okay, so I am leaving now. All right, from my side. Okay.
Okay, sir. See you soon.
Thank you. Thank you so much, everyone. Thank you.
Sandhya ma'am.
Yes, sir.
Sir has confirmed to join at the right time. So
Okay,
we
sir.
are waiting for this.
Okay, sir.
Announced to participant to be online. This is last session.
Dr. Avdesh Kumar Shirivastava will be joining soon. Kindly be connected, all the participants.
Sandhya ma'am, sir has joined or not?
No, sir have not joined yet.
Okay, he's trying to join. Good afternoon, Shirivastava sir.
Good afternoon, sir.
Sir, please,
Now we have with us Dr.
your voice is
Avdesh
not
Kumar.
audible.
Yes, sir.
Sandhya ma'am, please wait for a few minutes. Sir, your voice is not audible. Your mic is off, sir.
Now audible? Hello?
Yes, sir, yes.
Yes, sir.
Actually, your mic was off, sir. Good afternoon, sir. Welcome.
Thank you, sir.
Good afternoon, sir.
Sandhya ma'am. Sandhya ma'am, please start.
Now, yes, sir. Now we have with us Dr. Avdesh Kumar Shirivastava as our third speaker. I welcome Dr. Avdesh Kumar Shirivastava on behalf of Columbia Institute of Pharmacy, Raipur. Welcome, sir. Sir is presently working as Assistant Professor and HOD, Department of Microbiology, Government D.T.Post Graduate College, UTAI, Durg, Chhattisgarh. Sir has around 29 years of teaching experience. Sir is also member of editorial board in the proceedings of various national level seminars. Adding to his credits, Sir has around 57 research papers published in various national and international journals of repute. With these words, I would like to request Sir to deliver his deliberation.
Thank you, Sandhya ma'am.
Welcome, sir. Kindly start your presentation, sir.
Yes, ma'am. kindly tell me whether my slide is visible or not.
No, sir. The screen is not shared yet. It's still not shared, sir, from your side.
Okay, sir, I'm trying. Yes,
The
now,
slide is
sir.
visible, sir. Yes, sir. It's visible, sir. Kindly start your presentation.
Thank you very much. Uh, my This is my great pleasure to all of you. I'm Dr. Shirivastava. Uh, Actually, I have no words to share how I pay thanks to you because you have given a very good or for you, that is infection and disinfectant, and what is the world scenario behind it. First of all, we are very lucky enough, and we pay thanks to our God that He or they save us from the pandemic, which is just past few year back, or few months back, you better to say. After getting booster dose, everything now becomes normal. So a question comes in everyone's mind that what is infection or what should be the disinfectant? We have used various type of disinfectant. Not as a student of microbiology, many things has come in our mind. First of all, what is infection? So infection is any invasion of the body which is by any microorganism or parasite. And this severity is unknown because nowadays again we have suffering from flu-like symptoms. If you go to test, it becomes COVID positive or negative, but the symptoms are like same. But it is not so severe. But just we have passed a very important fatal condition past few months back, and I think in every family there is death in their entire uh, family members, and that was due to havoc of the COVID. So if infection is there, it may be due to bacteria, it may be due to virus or any agent. So we are using any chemical which can destroy, which can minimize the harmful effect of that microorganism can be regarded as disinfectant. So these disinfectants are the chemicals that destroy disease-causing pathogen or any other harmful microorganism. The pathogen may be bacteria, parasites, maybe rickettsia, maybe fungus, maybe virus, whatever it may be. So if we talk about infection, usually infection starts from a minor to a very major COVID-19, just we have seen. Confining myself to COVID-19, rather we are confining the total world scenario where we are suffering from such infection. It is either bacterial, viral, fungal, or any parasitic infections. The other parasites which is not included in this is either algae, species caused by Cephalosporus, that is red alga, Mycoplasma, actinomycetes, and they all can trigger sepsis. They all can trigger sepsis. That's why to save ourselves from the sepsis, from the infection, from their effects, we need certain disinfectant. This is the main topic for today's discussion. I pay my great privilege to Columbia University, entire faculty member, teachers, the senior professors, and their students. And we know that there are 7 causes of infection, And the common source of infection may be airborne, because just the scene has been passed. Airborne, that is COVID-19. Bloodborne, sometime, just recently a case I have observed in newspaper. A person was gone for dialysis, but unfortunately the blood which was transfused to him was unchecked, and he was suffered by HIV disease, and now he is no more. Sexually transmitted, we can talk about STDs, gonorrhea, syphilis, or any other thing. Fecal, so many microbes like algae, and fungus, bacteria, are transmitted by the fecal matter. Well, is also a very common source of infection. In environment, stagnant water is a very good source for Salmonella typhi. You may talk about bacterial dysentery, and warm water has also some tolerable thermoresistant organism. They can cause infection, and even by animal. We know that so many animals are there who are responsible for causing diseases. So these are the main sources of the infection. Disease. So we can see in this slide the person who is present inside of your hand. He is virus spreading respiratory tract. And whenever he breathes from nose, from mouth, some droplet like structures are emerging out. The droplets scattered zones are being from infected person in the form of either cough or droplets. And we maintaining two things: close your nose and mouth. And the second thing was maintain a social distance. So whenever we are present in the faucets area of the social distance, it may be any utensils, that may be any furniture, that may be towel, or even cloths. They may be contaminated, and airborne transmission is usually happens by less than 5 micrometers, and micrometer, and the droplets, which is 5 to 10 goes from land and to the surface. That's why they are not moving in the air. So the smallest droplets which are moving in the air is main source of the contamination. As far the disinfectants are concerned, disinfectants are disinfecting agent that is registered by the environmental agency. They are antimicrobial pesticides or those chemical. We have a long list of the chemicals. We'll proceed later on. They can control, they can prevent, they can destroy microbes. And we can divide these five categories. One is one. Biocides are also known as germicides. Whenever you the the term side means kill something. If you are we are saying like that biocides, germicides, side pesticide. These are the chemicals which can kill the microorganism. Actually, in general, it disinfectants as well as antiseptics as well as antibiotics. And what happens when we are using dermicides or herbicides? They generally react with the body protein, and especially the essential enzyme of the microorganism, and they may proceed for different force situations. Either they go for oxidation, or denaturation, or the substitution. That's why we are using a suffix side, biocide, bactericide, viruside, sporicide, such type of word is given. So there are two chances. One, either they may kill or they may give some hurdle growth. So when they are giving hurdle to the growth, they are known as static, like bacteriostatic or bacterial statistics, or bacterial growth can be inhibited, checked, virostatic, sporostatic, and these terms are added to the microbiome, which grow merely or inhibited or prevented from the multiplying. That's why the term biocides or germicide is there. Uh, all of you have kept this sanitizer in your box, in your pocket, even in your bag. So it is very interesting to say what is sanitizer. Sanitizer destroy or eliminate all bacteria, because suppose that if there is the viroid particle, viroids are those which are made up of only nucleic acid, and if you're using sanitizer, they will do not play any role, because they reduce the number of microorganisms on inanimate surface, like table, like your clothes, like your gloves, like your pen. These are inanimate surface, and they reduce the number of microbial contamination on those surfaces to level that are considered safe from the public health or standpoint. That's why we are using different type of chemical to to sanitize our room, sanitize our car, everything. And they may be formulation. There are a formulation of detergent and disinfectant. What is disinfectant? Disinfectant describe a product applied directly to an animate object. Whatever is present with us, this disinfectant is directly applied to them, and it destroy or irreversibly activate the panic microorganism. Some viruses, but not usually. Why we are using sanitizer for prevention of the COVID-19? Because the outermost layer was fatty acid, peplomers, the lipids was present. That's why they are known as crown organism. So the crown organism has outer lipid layer, and only due to presence of this organic solvent, we are using 60% alcohol to wash out that organism. And in absence of that protective crown, the COVID-19 was not in position to reinfect. That's why we are using sanitizer in usual mass. Okay. Antibiotics are applied to the surface of living organism or tissue to prevent or stop the growth of microorganism by inhibiting the organism or by destroying it. Now again, question comes: what is sterilization? All of we, the microbiology students, are using different types of sterilization, physical, like chemical. In physical, we are using laminar airflow, hot air oven, where we are utilizing the extreme heat. So the chemical like ethylene oxide, and they can these are the different process of sterilization, especially the market. There are a lot of agents which are regarded as detergents. Detergent is a big term which consider all the chemicals which can disperse or remove the soil or organic material from the surface. That's why they are allowing disinfectant to reach and destroy microbes within or beneath the dirt. And this is a very important that whenever you're culturing any microbes in our laboratory, the glasswares are usually washed by the help of detergent. These products are that is detergents. They reduce the surface tension. Surface tension is became a familiar word to all of you. To minimize the area and increases the penetration ability of water, so allowing more organic matter from the surface. Basically, the detergents are classified into three categories. One is cation, another is anionic, and another is non-ionic. As per quaternary ammonium compounds are concerned, they are, in short, they are used QAC. They're a very good uh, agent, and the cationic detergents are positively charged, with the exception or quaternary are seldom used in cleaning ingredient. Second is anionic detergent like soap, the salt or fatty acids. They are less ideal for that may allow soil or microorganism to accumulate. That's why usually we are using this, but we can. Next and last is a non-ionic detergent. They are very good emulsifier. Emulsifier means the separation of the fat molecule or the lipid molecule, penetration or dispersion, effective at lowering surface tensor and have reduced foaming properties. That's why these products do not typically complex with metal ion, and such as those found in hard water. So most commercial detergents are a combination of anionic and non-ionic. We are using common mixture form. Next is physical disinfection. Since a very ancient time, we are worshipping the sun, and sunlight as well as light, as well as the energy which is coming to our earth is electromagnetic radiation. So these are the three very physical disinfectant. Recently, we have celebrated Ozone Day. We are now in position to say that they may be used to reduce or eliminate microorganisms from the environment. The use of heat is one of the oldest physical control against microorganisms and are fairly reliable method of sterilization. That's why so many crops, so many vegetables, so many uh, I mean edible materials are also being put in sunlight so that they can be saved. So sunlight has also a very important agent, important ingredient, that is UV light. So UV light have a detrimental effect on number of microorganisms, and same thing we are providing in our laboratory by HEPA flow, and that is laminar airflow. And even in a small culture room, we are using UV light. So they form dimer of the microorganism in their DNA. So sunlight and ultraviolet ray had detrimental effect on a number of microorganisms, may be practical method for inactivating virus, mycoplasma, bacteria, even fungi. Particularly those are airborne. So UV light sterilization capabilities surface because it's of penetrating power. And nowadays we are also using microwaves, gamma radiation, and freezing is not very reliable method, but they can increases the shelf life of the food material so that we can keep this food materials in our refrigerator for a longer time. But there are certain microorganisms which are cryophilic, which are cryogenic, which are freezer lovers. That is, they can survive in low temperature. So they are also being used here. As for the chemicals are concerned, uh, each chemical has a unique characteristic, hazards, toxicity, efficacy against various microorganisms. And environmental condition is very important because presence of organic matter, the pH, the water hardness also give a very important impact on the disinfectant. Now come to first chemical, that is acids. Usually in laboratory we are using acetic acid or citric acid. And what is the role of the acid? Acid disinfectant function to destroy the bond of nucleic acid. And if nucleic acid bonds are destroyed, naturally DNA or gene or the chromosomes or the main part of the organism is being destroyed. So they are not in position to do or perform their pathogenic effect. This is one of the logic. Another logic is the protein are getting precipitated. So if proteins are getting precipitated by the help of acid, so your enzyme becomes denatured or inactivated. So these are the two important things. We can retard or we can remove the infections. So acid also changes the pH because we have studied strong acid, soft acids, so the different pH, and pH is very important for the environment of the microorganism which they are growing. Even in culture media, we are performing different activity for maintaining the pH. pH is also very important, and they are being maintained by different buffers, and acid can be also used. And concentrated solution of acid can be caustic because it may causes chemical burn, and that can be toxic at high concentration in the air. Next is acetic acid. The acetic acid which is sold in the market are usually 95% acetic acid. They are known as glacial acetic acid, and they are usually diluted to the 5%. And concentrated form of the acetic acid is corrosive to the skin and lungs, but typical dilute 5% is considered as non-toxic and non-irritating. That's why acetic acid is typically applied for spraying, misting, emerging, in a diluted form. Household vinegar, which we are usually we are eating in our fast food for sourness of the food, it may be chow mein or even pasta. The 4 to 5% solution of acetic acid by volume, and acetic acid is a poor activity in organic material. So the fast food can be left for a longer time due to because it disinfect. Another chemical is alcohol. Alcohol may be used either in the form of ethanol, the ethanol that is ethyl alcohol, C2H5OH, or isopropanol. Um, both these alcohol are very common. And alcohol has a broad spectrum antimicrobial agent that damage microorganisms by denaturing proteins, causing membrane damage and cell lysis because they are very much lover of the water. They can take water, and due to endosmosis, cell lysis, membrane break might be possible. And alcohol is also very good solvent because the plasma membrane of the organism, which is made up of lipoprotein, so they may be soluble in alcohol. That's why in our COVID infection, we are using 60% alcohol should be there, so that alcohol can be used as surface infections, topical antiseptic, and hand sanitizing lotion. Alcohol are considered as fast-acting, capable of killing most bacteria within 5 minutes to exposure, but are limit to ineffective against spores because the nature is solubility. Ethanol is considered as biocidal, but just I was discussing the same thing just little moment before. That is, isopropyl alcohol is not effective against non-enveloped viruses. That's what the crown structure, that is papillomere, that is lipid layer that was present in COVID-19 was effective for alcohol. An important consideration with alcohol is the concentration used, with 70 to 90% being optimum, and even higher concentrators, that is 95% is usually. See, alcohol is effective, but when it is present in higher concentration, it is less effective. And this less effect is perhaps due to some degree of water required for efficacy, because normal alcohol do not perform their efficiency. That's why we are using the water to make it more efficient, and they denature the proteins. Uh, a very important logic behind alcohol is alcohol evaporates more quickly, and whenever you're using alcohol, alcohol is evaporated and nothing is behind. So no residue should be there. This is very important. I mean, landmark. I mean, category or characteristic of the alcohol. So the activity of alcohol is limited to the presence of organic matter, and we know that alcohol is very inflammable, and they can be kept, or they can glass, because rubber and plastics, they are corrosive for them. If we are getting I mean, direct injury or irritating at the skin, they give a type of surface sterilization. As for the aldehyde group is another group. Usually we are using two type of aldehyde, formaldehyde and glutaraldehyde. So aldehyde are very effective broad spectrum disinfectant because it denature the protein and disrupting the nucleic acid. These two property are very common to most of the chemicals, and that's why the aldehyde is very agent against bacteria, fungi, virus, mycoplasma, or even spores of different organisms. So aldehyde are non-corrosive to metal, rubber, plastic, and cement. That's why they can be kept in this container. These chemicals are highly irritating, toxic, and human or animals with contact or inhalation are potentially carcinogenic. Therefore, their use is very limited. Uh, one tour was organized by my students, and when we are going for the botanical collection, formaldehyde was present for preservative, and some bottle was broken. 1984, when the Bhopal gas study was just happened, total train was evacuated because the peoples are the irritation of the formaldehyde was such that they think that something has leaked in the compartment, and total compartment was vacated, and at half an hour, about half an hour, the train was standing on the station. That I usually remember when this aldehyde comes in our mind. So if we think about nitrile gloves, the fluid-resistant gowns, PPE, the personal protective equipment that was used by the doctors of COVID-19 uh, patients, and they are also sterilized in this, should be worn if using these chemicals. Formaldehyde and glutaraldehyde, these are the two very important. So formaldehyde is used for surface infectant and fumigants, has been used in decontaminate wooden surface, bricks, uh, cervix, crevice, or electronic and mechanical equipment. And usually we are leaving this 24-hour for surface. Uh, that's why the biological equipments, biological museum specimen are usually kept in this formaldehyde solution. And it is highly volatile. Just I have told the story. So at least the closure should be 25 hours after treatment. The efficacy of formaldehyde is dependent on relative humidity and temperature. Optimum being humidity close to the 70 and temperature closest to 50. Actually, this is not zero. This is degree Fahrenheit. And some this was my fault that I cannot make it 70. So please make a correction here. So formalin is 70% solution in formaldehyde in water. So and another is glutaraldehyde. This is primarily used as disinfectant for medical equipment like endoscopy, rectinoscopy, endoscopy. They are the very important equipment for our elementary canals study. Inner layer of the elementary canal can be visualized by endoscope. So this glutaraldehyde is usually kept in that. Whenever you go to the endoscopic doctors, they are keeping their total tubes in this and can be provided sterilization for a prolonged contact times because they remove their endoscopic machine from one intestine to another. That's why the 2% concentration is considered as high level of disinfection. Its efficacy is highly dependent on pH and temperature. Working best at a pH greater than 7 and high temperature, it considers more efficacious in the presence of organic matter, soaps, and hard water than formaldehyde. And this Sidex is now available in the market, which is used for surface sterilization. Next is few alkalis like sodium or ammonium hydroxide, sodium carbonate, calcium oxide. And we know that alkaline agents work for saponifying lipid within the envelope of microorganism. This activity of alkali compound is slow, but can be increased by raising the temperature. Alkali had a good microbiocidal properties, and they are very corrosive agent, and personal protection precautions should be observed whenever using the alkali. The sodium hydroxide is very, very common, which is also known as lye, caustic soda, soda ash, or a strong alkali, and it is used for disinfect building, but is highly caustic. Protective clothing, rubber gloves, safety glasses should be worn whenever using chemicals. Lye should be always be careful and added to water. Otherwise, it may give something harm to you. Never pour water into lye. A very violent reaction will occur, as well as the production of high heat can melt the plastic container. That's why we are usually um, keeping this process in a metal container. Sodium hydroxide is very corrosive for the metal, uh, so it is considered as effective FMD. In fact, FMD is the foot mouth disease. The very common disease in cattle, known as Khoraha. In Chhattisgarh, it is known as Khoraha. The cattle are died due to foot and mouth disease, FMD disease, and that is infected disease. Next is ammonium hydroxide. Is infected against toxicidal oocytes, have a strong solution emit intense and pungent fumes. The substance is not considered effective against most of the bacteria. Uh, Next is sodium bicarbonate, which is also known as soda ash or washing soda. Sodium carbonate, that is soda ash or washing soda, has been used in a hot solution, that is 180 degrees Fahrenheit. The term is Fahrenheit. You can change this into Celsius also. Uh, by disinfecting building which have the house animals with FMD, because wherever foot and mouth disease viruses are, infection occurs, the total entire cattle colony, the total bunch of the colony of the cattle are being infected by this, and most of them are die. That's why we are using this sodium bicarbonate for the surface sterilization, so that if once it comes to quarantine and other processes, also very effective one. It is more effective as a cleaner than disinfectant, since it lacks efficacy against some bacteria and most of the viruses. A 4% solution has been listed as an approved chemical for FMD virus. It has poor activity in presence of organic material and can be deactivated by the hot water. And last is, it can be irritating and require protective clothes and is harmful to aquatic life. That's why whenever the total disinfectant is carried, and if you're pouring this water inside the lake or any other water reservoir or water tank, they usually contaminate, or they kills the life, they disturb the life of the pond. So we should be very careful. Integrated pest management applied should be there, so that we can save the life of others. Because once you are saving the life of cattle, simultaneously you're killing the fishes of the pond. So what type of management or biodiversity conservation is this? I have no words. Next is calcium oxide, that is also known as quicklime. Calcium oxide, that is lime wash, is mixed with water. And perhaps I think every year in our old, when we are very young child, we have seen that our parents are using this calcium oxide to clean our different surfaces of our house. The different wall are being stained by calcium oxide quicklime. This is biocidal effect, and some bacteria and viruses, and in sometimes sprayed on the ground following depopulation of infected termites, and have also been used to retard and putrefaction or buried carcasses after depopulation. And that's why nowadays we are also using to remove algae contamination, fungal contamination on the surface. And it is not very effective against FMD virus. The common mouthwash, that is chlorhexidine, which is scientifically known as bigonides. So bigonides, you can get this chlorhexidine, very common mouthwash. So these bigonides are detrimental to microorganisms by reacting with the negative charge group of the all cell membrane and alters the permeability. So if permeability of the microorganism is being changed, so you can say what will happen to that. So the bigonides have a broad spectrum antibacterial activity, and they are limited to very less effectiveness against viruses, and not very sporocidal, myobacterial-cidal, or fungicidal. That's why the bigonides only function at limited pH, that is 5 to 7. So our mouth pH is also very similar to that. That's why it is very effective in our mouthwash, and they are easily inactivated by soap and detergent. Because in mouth you cannot put your soap and detergent, so chlorhexidine is usually used to disinfect our mouths. These products are toxic to fish and should be discharged into the environment. Halogen, we know that halo compounds, chlorine, iodine. Halogen compounds are broad spectrum compounds that are considered low toxicity, low cost, and easy to use. They do not lose potency over time, and they are very effective, and they can survive even up to temperature 110 degree Fahrenheit, and pH is 9. Since these compounds lose activity quickly in presence of organic debris, sunlight, and some metals, because you know that chloroform is always kept in bottles, because and oxygen, they form phosphogen type of dangerous gases. So this applied on clean surface for disinfectants. The chlorine compounds function through electronegative nature and denature protein, and are considered broad spectrum, being effective against bacteria, enveloped and non-enveloped viruses, mycobacteria, and fungi. At elevated concentration, chlorine compound can be sporified. This is very common, sodium hypochlorite. During COVID-19, we have washed our, carried over, we have washed our railings by the help of sodium hypochlorite. It is one of the most widely used chlorine-containing disinfectant, commercially chlorine-based containing 5.25 sodium hypochlorite in aqueous solution and 550,000 ppm available chlorine. So this is biocidal activities determined by the amount of chlorine that is present in the solution. Low concentration are very active against vegetative bacteria, fungi, and most of the viruses. Due to only this logic, during this infection pandemic, we have used sodium hypochlorite. And its rapid sporicidal action can be obtained around 2,500 ppm parts per million. However, this concentration is very corrosive, so it should be limited to its use. High concentration is also very irritating to the mucous membrane, eyes, and skin. That's why usually whenever we are sterilizing our people who are working on them are do not liking to work by this, and they're protecting their eyes and other things, and mouths. So chlorine compounds are readily uh, deactivated by light and some metal, and so fresh solutions should be always used. And hypochlorite should never be mixed with acid or ammonia, because if it is mixed by them, again chlorine gas will be eliminated, and that becomes very toxic for all of us. Next is iodine. Iodine compounds are broad spectrum and considered effective for a variety of microorganisms like bacteria, mycoplasma, fungi, viruses, and they also denature the protein and interfere with the enzymatic system of the microorganism. And iodine are often formulated with soaps and considered relatively safe. And concentrated iodine compound may be irritating to the skin, can or damage rubber and some metals. That's why in microbiology lab, usually we are using iodine solution for staining bacteria. We are wearing the uh, I mean protective coats for this. And iod force are another iodine complex that has been increased solubility and sustained release of iodine. Of common use iodophore is given by povidone iodine. That is a very common, surface spray, because whenever you cut, you are usually using this povidone iodine on your surface, so that they can, because whenever any cut burn occurs in our body, this povidone iodine actually inhibits the growth of other microorganisms, so they cannot come in our bloodstream. They are good in general use and less readily inactivated by organic matter than elemental iodine. The dilution of iodophore actually increases the free iodine concentration and antimicrobial activity. Um, Ultimately, we are coming to oxidizing agent, hydrogen peroxide, paracetic acid, or vircones. Oxidizing agent are peroxide-based compound that function by denaturing the protein and lipid of the microorganism. And peroxygen compounds vary in their microbiocidal range. They are considered effective on hard surface and equipment. In their diluted form, these agents are relatively irritating or damaging the clothes when concentrated. That's why usually the compounder, whenever washing your wounds, are using this hydrogen peroxide. And this hydrogen peroxide uh, is home in the home is diluted to 3 to 10%, whereas industrial use it is concentration may reaches up to 30%. Hydrogen peroxide is very effective in between 5 to 20%. And in this concentration, they are considered as bactericidal, virucidal, and virucidal non-enveloped viruses may be resistant, because actually they oxidize and nothing is present. It should be a non-enveloped, or so they have no role. Virucidal at high concentration is sporicidal. Its activity against mycobacterium is limited. Next is parasitic acid. Uh, it is also a very strong oxidizing agent, and they are hydrogen peroxide and acetic acid. It is also considered as bactericidal, fungicidal, sporicidal, virucidal, and it is also very effective against mycobacterium and algae and some activity of the presence of organic material. And last, in market, Vicon is also available in the form of potassium peroxy monosulfate or sodium chloride. It is peroxygen molecule organic acid and surface combination, combination, and with a wide microbial spectrum of activity and some efficacy in the presence of organic material. The last but few is phenol, and in market is a tetrol, phenotic, like name it is available. Phenol are broad spectrum disinfectant that function by denaturing protein and inactivating membrane bound enzyme like permease and alter the cell wall permeability of the microorganism. The cell wall is dead and permeable in most of the cases, but at the composition of the cell wall changes, it also changes. Phenol can be coal tar derivative or synthetic formulation and usually have a milky or cloudy appearance when added to water, and as well as in a strong pine odor in toilets, in our houses. Usually phenyl is being used. So pine sol is example of phenol found in the home, that is phenyl. Phenols are typically formulated in soap solution and increases their penetrative powers. So is like this skin infection when occurs, usually doctors prescribe this, and at 5% concentration they are considered as bactericidal, tuberculocidal, fungicidal, and virucidal for enveloped viruses. Phenol are not effective against non-enveloped viruses or spores. Phenol do maintain activity in hot water and presence of organic matter and have some residual activity after drying. And phenolic disinfectants are generally safe for human, but prolonged exposure to the skin may causes irritation, and concentration over 2% are highly toxic to the animals, especially the cats. The QAC, that is quaternary ammonium compound, in market it is known by Rocal, Jefferin, Diquat, D256 or quartz. So these compounds are cationic detergent that attract natively charged surface of microorganism. And we know that plasma membrane has negative charge. That's why they are very important for gathering the negatively charged plasma membrane towards them, where they irreversibly bind phospholipid, and that phospholipid was present in the plasma membrane and denature the protein, impairing permeability. As per the QSC can form different generation depending upon their chemistry. And nowadays it has germicidal, less foaming, and more tolerance to organic load is seen. And this is highly effective against gram-positive bacteria, good efficacy against gram-negative bacteria, fungi, vellum viruses. They are not effective against non-enveloped viruses, mycobacterium, and QSC have some residual effect keeping surface bacteriostatic, and they are more active at neutral or slightly alkaline pH and less in 3.5 pH. It is very stable in storage, but generally it is highly in hard water, and it also depends upon the generations. Uh, this is very toxic to fish. That's why they should not be uh, directly given in water sources like pond, lakes, streams. And the last agent is plasma. I think this is not a very rare term to all of you. He was Irving Langmuir, who has in 1927 termed the word plasma. And plasma reduces concentration of the pathogen whenever contact with contaminated surface. So this plasma can destroy bacteria, virus, fungi, or even prions. Prions are only made up of protein. There is no any other things. Rusner has reported prions. It also removes toxins and further organic contaminants. Even complete sterilization is also possible due to prolonged exposure of plasma. This is very costly because early application of plasma technology was used only in field of engineering like nuclear fusion, plasma etching. But nowadays, since last 20 years, uh, so many things has been given microbiocidal properties of the plasma, and recently this accumulated knowledge use this plasma technology in disinfections as well as in sterilization, so we can use the potential use of this plasma. what is plasma? A question comes in our mind. So we, all of you, I think, is familiar with this term. This is nothing new for all of us. We know that solid, liquid, gas are the three common stages of the matter. And when solid is heated, it is converted to liquid. And when liquid is heated, it is converted to plasma. It is converted to gas. And when more energy is applied to the gas, they becomes ionized. And this ionized gas is known as plasma. And this can be considered as the fourth fundamental stages of the matter. So the plasma has reactive chemical species like electrons, ion, neutral molecules, or atoms, as well as some charged species. So in addition, emission of radiation, that is UV, is as well as visible and near infrared region during the plasma generation. So this is now very much used, and this is the machine which can be used for plasma generation. And if you use this plasma, uh, the direct touch can be eliminated, because you can see in this diagram the switch is there and we are giving plasma dose on this switch, and the switch becomes uh, disinfectant. So the whatever microbes was there, they are eliminated by the help of plasma. So very soon we will getting this type of sterilization process and disinfectant process in our daily life. These are uses which I am gathered, and again, God save all of us so survive on this earth. And the charming face of this lady says that she has no worry of anything. Even if we put few feathers on our head, by traditional method we can survive. And this is the pleasure of our Chhattisgarh people, that every sarve bhavanti sukhina Sarve Bhavantu Niramaya. So these are the things which I want to share. Infection is there, but if you are ready for the disinfectant, we have no worry. We can save ourselves by the infections. And thank you very much to the organizer, to the convener, to the secretary who has given me to this opportunity to share few things with you, the you learner people. So I'm very much, I think I'm lucky enough to share my opinion before you. If we have any query, please ask.
Participants may ask any question if they have. Thank you, sir. Thank you so much for sharing your profound knowledge with us.
Thank you, ma'am. And thanks to all of you.
Yes, sir.